In the previous video lecture, we covered the transformation experiment that was conducted by Frederick Griffith in 1928 and which laid the foundation of the Avery, MacLeod, and McCarthy experiment conducted in 1944 to prove that it is DNA which is acting as a genetic material and not proteins. Now, in this video lecture, we will be covering the Hershey and Chase experiment, which confirmed that it is DNA which is acting as a genetic material and not the proteins. So, the Hershey and Chase experiment, also known as a Blender's experiment, was conducted by Alfred D. Hershey and Martha J. Chase in 1952, and this experiment confirmed that it is DNA which is acting as a genetic material and not proteins. Now, additional direct evidence indicating that DNA is a genetic material was published in 1952 by experiment conducted by Alfred D. Hershey and Martha J. Chase. These experiments showed that the genetic information of a particular bacterial virus, that was bacteriophage T2 with which they worked, was present in the DNA. The natural host for T2 phages is the bacterium Escherichia coli. Now, Hershey and Chase had to demonstrate that the DNA injected by the phage particle into the bacterium contains all the information required to synthesize progeny phage particles. Now, before the 1952 experiment of Hershey and Chase, the reproduction cycle of bacteriophage in E. coli cell was known. So it was known that whatever enters into the bacterial cell is carrying all the genetic information, and it is capable of passing on this genetic information to the next generation. So whatever is carrying all the genetic information and is capable of passing on the genetic information, we know is the genetic material. So what Hershey and Chase had to prove was that what is entering into the Escherichia coli from T2. That is, if protein is entering into the bacterial cell, then protein is carrying all the genetic information and is capable of transmitting that information to the next generation. If DNA is entering into the bacterial cell, then DNA is carrying all the genetic information and that is capable of passing it to the next generation also. That is, it is capable of forming the progeny phage particles. The advantage with using T2 phage is that it's a very simple structure. It is composed only of protein and DNA in a ratio of about 1:1. So that is, it is composed of 50% protein and about 50% DNA. So Hershey and Chase demonstrated that it is the DNA of the virus particle that enters into the host, whereas most of the protein of the virus remained adsorbed to the outside of the cell or is present as a supernatant when centrifuged. This strongly implied that the genetic information necessary for viral reproduction was present in the DNA. The basis of the Hershey-Chase experiment is that DNA contains phosphorus, but it has no sulfur, whereas proteins contain sulfur but has no phosphorus. So a single particle of T2 phage contains DNA, which is a phosphorus containing substance encased in a protein capsid, in a coat, in a shell of protein, and that protein contain amino acids methionine and cysteine, which contain sulfur. So they were able to specifically label the DNA of the phage as well as label the protein of the phage by setting up two experiments. Number one, by growing phages in a nutrient medium in which radioactive phosphate, that is, the radioactive isotope of phosphorus, P32, was present, and that was the only source of phosphorus. Therefore, phages containing radioactive DNA could be prepared. In the second set of experiment, medium contained radioactive sulfur, that was S35, in place of the normal isotope, that is S32. So phages containing radioactive proteins could be obtained. Now, in these two labeled phages, one labeled on the DNA and other labeled on the protein, was separately used to infect the host, that was E. coli. Phage DNA and the phage protein coat could always be located because of their radioactivity. So using these two labeled P32 as well as H35 labeled DNA and protein respectively, Hershey and Chase could show that P32 is injected into the host, that is the bacterium, and S35 mostly remains outside of the bacterium in the supernatant or adsorbed onto the bacterial cell surface. The T2 phages labeled with S35 were mixed with the host, that was E. coli. T2 phages, they attach to the host and then they inject the genetic material into the host, and the attached phage can be torn from the bacterial cell surface by violent agitation using a blender. So that was done. T2 phages in one set of experiments labeled with the protein on S35 mixed with the host, that was Escherichia coli. After a few minutes was centrifuged in a Waring blender. It was found that most of the radioactivity could be removed from the bacterial cell, and that did not affect the progeny production. That means most of the radioactive sulfur remained outside the bacterial cell in the supernatant, or some attached to the bacterial cell surface, did not enter into the bacterial cell, but progeny was still produced. Similarly, when the T2 phage is labeled with P32, that was on the DNA labeling, was mixed with E. coli for a few minutes and then subjected to centrifugation in a blender, it was found that radioactivity was found inside the cell. That is, it was not removed by blending, it was not removed by centrifugation. So because radioactivity was found inside the host cell in the case P32 label was used, that means where DNA was labeled, that means DNA was entering inside the E. coli cell, and protein in the first set of experiment remained mostly outside of the cell. So this picture shows the two experiments which were conducted. In the first experiment, what was labeled is the protein. That means S35 or sulfur 35, which was labeled for protein, that was used. And second set of experiment shows the label for the DNA. That is P32, that is a labeled DNA experiment. So in the first set of experiment, S35 labeled phage particles were adsorbed to the bacteria for a few minutes. After centrifugation and blending, the pellet, which contained bacteria, and supernatant was separated. It was found that 80% of the S35 was present in the supernatant and 20% was found in the pellet. Later, this 20% was shown to be those phage tail fragments which were too tightly bonded to the host cell surface so that they were not removed by blending. In the second set of experiment, in experiment 2, P32 labeled phage was used. That means labeling was on the DNA. And what was found was that 70% of P32 remained associated with the bacteria and therefore settled down as pellet when it was centrifuged. 30% of it remained in the supernatant, which was later shown to be that phage particle which during blending broke when it was still being transferred into the host cell, when it was still being injected into the host cell, it broke during blending and came in the supernatant. Some was found because of they were defective phage particles not capable of injecting their DNA into the cell. So this experiment proved that radioactivity, if it was present on the DNA, could be seen inside the host cell, inside the E. coli cell, and all the phage progeny that was generated also had the P32 labeled phages. In the first experiment, protein was labeled. Most of the radioactivity shown in green here was found in the supernatant. The progeny phage particle was still produced as such. There was no change. And by this experiment, Hershey and Chase showed that it is the DNA which is entering into the cell. Protein remains outside the cell. So whatever is entering into the cell is the genetic material. It is carrying all the genetic information and it is capable of passing on that information to the progeny or to the next generation. So this was a confirmatory experiment to prove that DNA and not proteins are the genetic material. Now, wherever DNA is not present, which is mostly in the case of RNA containing viruses, where DNA is not present, RNA is present, RNA will be acting as the genetic material. So here also, proteins will not act as a genetic material. If DNA is not present, then RNA acts as the genetic material. So there was an experiment conducted to prove that RNA acts as a genetic material. The experiment was performed by Frankel Conrad, Stanley, and Sanger in 1957, and the experiment was done on virus that was a tobacco mosaic virus. Now, this tobacco mosaic virus also has a very simple structure. It has RNA and it has a protein coat. So there is only RNA and there is protein coat. This tobacco mosaic virus particle, it was collected from the infected tobacco leaves, and by chemical treatment, protein coat of tobacco mosaic virus was separated from the RNA. So they were separated from each other by gentle detergent treatment using SDS at neutrality, and the RNA was then collected with ammonium sulfate precipitation. The protein was separated from RNA. Cell debris, when it was introduced into tobacco leaves, the leaves remained healthy. But when the cell filtrate was introduced into tobacco leaves, it was infected with the virus and it died. They further took this experiment to the second level, to the second step, and called that experiment as the reconstitution experiment. Now, for the reconstitution experiment, they took two different strains. One was tobacco mosaic virus or TMV, and second was Holmes ribgrass virus, that is HRV. Now, HRV is a variant of TMV. It has a different amino acid composition. The strains differ from each other in their amino acid composition, and histidine and methionine they are commonly not present in tobacco mosaic virus, but they are present in the Holmes ribgrass virus. So they took two different strains. One was TMV, the other was HRV, and from both the strains, they separated the RNA from the protein coat. Then what they did was they produced the hybrid viruses. They mixed them. That means RNA from one strain mixed with the protein coat of the other strain and vice versa. So reconstituted viruses were made by mixing proteins of one strain with RNA of the second strain and vice versa. When these mixed viruses were infected to the tobacco leaves, it was always found that the progeny, phenotypically as well as genotypically, was identical to the parent from where RNA has been obtained. So the phenotype and genotype of the progeny always resembled the parent from which RNA was taken and not from which the protein was taken. So this is a TMV particle, this is a HRV particle. Both have a simple structure. There is a RNA filament and there is a filamentous protein. The RNA and proteins are separated by chemical treatment. Of the TMV, the RNA and proteins of the HRV are also separated using chemical treatment. Then a reconstituted or a hybrid virus was generated which contained RNA from TMV and protein code from HRV. This reconstituted virus was then made to infect tobacco leaves. The progeny particle phenotypically as well as genotypically. How do we know phenotypically? TMV causes a mottling disease on the tobacco leaves. HRV causes ring formation. So if you're taking RNA from TMV, the disease caused will be mottling. Lesions will be formed. If you are taking RNA from HRV, the disease caused will be ring shaped. So phenotypically, as well as when the progeny is studied, genotypically also, the progeny resembled TMV particles completely. That means the RNA was also of TMV and the protein coat was also from TMV. There was no part of the HRV in the progeny viruses. So the progeny viruses reflect the source of RNA which was taken. So if we take the TMV RNA, disease caused will be mottling. The progeny will resemble TMV completely. If we take RNA from HRV, the disease caused is ring shaped and the progeny will resemble HRV completely. So by this experiment, it was concluded in 1957 that if DNA is not present, then also proteins do not act as a genetic material. It is the RNA which will act as the genetic material. Thank you.